So thank you, everyone. My name is Lauren Sheehan. I'm a graduate student from Virginia Tech, USA. And before I begin, I wanted to thank the Brucellosis Committee for accepting my abstract for this talk. It's a great honor, and I'm excited to talk to you guys about my project. So I'll be talking to you about a regulator that I'm studying that we found to be very important in the pathogenesis of Brucellosis. And moreover, we found that it's very conserved amongst alpha proteobacteria. So I thought that's very interesting, and I'll be talking about that later. So just a brief introduction to this regulator. It's a lysotype transcriptional regulator, so it's the most common prokaryote regulator, and it controls a variety of genes. So genes involved with virulence, form sensing, metabolism, and it can either act by activating or repressing gene transcription. And as a family, these regulators are very conserved in the structure. So they have a C-terminal co-inducer binding domain and then an N-terminal DNA binding domain. And what makes them different from each other is normally the co-inducer binding domain, so what they're actually responding to. And I'm going to be switching a little bit here. It's going to make sense why I'm going from regulator to small RNAs. Felix talked about small RNAs yesterday, so I'll make this quick. So they're usually between 50 and 300 nucleotides, and they act by base pairing with their target mRNA, usually imperfectly with the help of HFQ. And they can also modulate protein activity. Two small RNAs that we're interested in our lab are ABCR1 and ABCR2. And ABCR1 and ABCR2 regulate different ABC transport subsomes within Brucella. And we also found that they're related to virulence. So in regards to ABCR1 and ABCR2, what was found, and it's very interesting, is that they seem to be performing redundant functions of each other. So if you have an isogenic mutant of ABCR1 or an isogenic mutant of ABCR2, you see no attenuation compared to 2308. You only see attenuation when you have a double mutant. So again, this is showing that they have redundant functions of each other. So the question that we had was, what's regulating these small RNAs? And if you look at chromosome 1, flanking ABCR2 is a lysar gene. And I didn't mention this before, but lysar genes are known to regulate genes within their vicinity. So this was a prime candidate for potentially regulating ABCR1 and ABCR2. So a mutant was made, and northern blot shows that this lysar gene is controlling expression of ABCR2, which is interesting, and specifically ABCR2 and not ABCR1. And moreover, an MSIT was done to show that this lysar regulator is indeed binding to the small RNA, and to show that it's specific to. So ABCR1 is over here, and it isn't binding, so it's specific to ABCR2. And based on the results that we saw with ABCR1 and ABCR2 involved in pathogenesis of Brucella, we want to see how this regulator is involved as well. As you can see, the red line here is our lysar mutant, and it is attenuated compared to 2308. Moreover, we want to see how this lysar regulator is involved with a chronic infection within mice. And again, the red bar is our lysar mutant, and as you can see, it is indeed involved with virulence in Brucella. So the next two slides are just showing complementation. We want to make sure that the attenuation that we were seeing was from the lack of this regulator. So I'll just draw your attention to this orange line. This is our complement, very similar to 2308. And then for spleen colonization, we also want to make sure that the attenuation was because of lack of this regulator. And as you can see with our complement, very similar to our wild type 2308. So after calling this the lysar gene for a while, we finally came up with a name for it, and we named it VTLR for Virulence-Associated Transcriptional Lysar Family Regulator. And so like I said before, I'm from Virginia Tech, so I was able to incorporate the name VT into naming the regulator. So this is a picture of Burrs Hall, which is our main campus building. So one of the interesting questions that we had is, we see this attenuation with ABCR1 and ABCR2 only when you have a double mutant. And the question we had is, why are we seeing the attenuation for our lysar regulator? Because technically this regulator is only controlling expression of ABCR2. So it didn't really make sense, so what we did next was a microarray to see what else is this regulator controlling. As you can see on the top, there are 3 genes, 3 small hypothetical proteins that are also being activated by this regulator. But I'm not going to get into that. My lab mate, James Budnick, he has a poster, and his project is basically characterizing these hypothetical proteins. Not a lot is known about them, and it's a really interesting project. So if you have any questions, his poster is number 32, and I suggest that you all take a peek at it. So my project's more looking at this regulator and characterizing this regulator. So based on the microwave, we want to make sure that this regulator is controlling expression of these hypothetical proteins. So we did a northern blot, and as you can see, the wording kind of messed up here. But in our mutant, you don't see expression of these hypothetical proteins. So next, what we did is we looked around the bindings, we looked around the promoter regions of these 4 genes, and we found upstream of the -35 a conserved sequence found in all 4 VTLR regulated genes. So we think this is where VTLR is binding to. Further experiments need to be done to confirm this, but it is very interesting. And what also kind of was really interesting is in alpha proteobacteria, if you look at these hypothetical proteins, in some of the promoter regions, this sequence is there too. So again, further testing to be done to confirm this is actually the binding site, but thought it was pretty cool. So this is a picture from a site called Microbes Online, and this arrow is pointing to VTLR, which is up here in Brucellosis, and these are all different alpha proteobacteria orthologs of VTLR. So you can see that this regulator is very conserved. So it kind of draws the question as to if this regulator is potentially a global regulator in alpha proteobacteria. Not a lot of experiments have been done in other bacteria so far, so it'll be interesting to see what this regulator's role is in other bacteria. So in the beginning, when I was talking about these lesser-type transcriptional regulators, I said that they're very conserved as far as their structure. The main difference is in their co-inducer binding domain. So we wanted to see exactly how similar they were and to see if these were actually true orthologs. So as you can see here, this is percent amino acid identity and similarity, and they are very, they seem to be true orthologs. So it is interesting to find this high conservation of this regulator. And lastly, and very briefly, I'll just talk about this one paper that was published by a Sinozobian group. And what they did is they looked at a number of lysar regulators, and they wanted to see which ones were involved with symbiosis. And lo and behold, they found one called LSRB that is an ortholog to VTLR that is involved with the symbiotic relationship of Sinozobia and alfalfa plants. So without this regulator, Sinozobia is not able to have that symbiotic relationship with alfalfa plants. So this is showing that these regulators are very important in bacteria-host interactions. So my project right now, what I'm moving towards is trying to characterize this regulator and figure out what is the co-inducer that it's binding to, how it's binding to DNA, and I guess transcribing the genes. So that's where I'm kind of heading. Hopefully next time in Chicago I'll be presenting that data, and it'll be very interesting. So very briefly, I wanted to thank Caswell Lab, my graduate committee, and then everyone else that has been involved with this project as far as mice and funding and sequencing. I'm very grateful. And then I'll open up the floor to questions, and thank you for your attention.